Evening, ladies and gentlemen. Karibuni sana. In this tutorial, we explore the 2000 KCSE Biological Sciences Practical, an essential guide for students preparing for Paper 3. We will walk through all three sections, from osmosis in potato cylinders to bone structure analysis and seed germination. This video is designed to help you understand the science behind the procedures and not just memorize them. Let's demystify the practical and build confidence for your exam success. Let us now look at the confidential. Question 1. 6 freshly obtained potato cylinders measuring exactly 4 centimeters. Use cork borer with a bore of 0.5 centimeters diameter. 20 ml distilled water in a 50 ml beaker labeled S1. 20 ml saturated sodium chloride solution in a 50 ml beaker labeled S2. Clear plastic ruler that can measure in centimeters, and blotting paper. Question 2 requires a scapula obtained from goat or sheep labeled specimen J, 2 pieces of ribs minimum length 5 centimeters labeled K1 and K2, soak K2 in 2 molar hydrochloric acid overnight. The ribs should preferably be obtained from goat or sheep. Blotting paper. Question 3. Confidential. Maize seed soaked overnight labeled specimen L. A bean seed soaked overnight labeled specimen M. And dropper, 2 petri dishes, scalpel, and a hand lens. Let's come to the questions. Question 1. You are provided with 6 potato cylinders and solutions labeled S1 and S2. A. Measure the length of each cylinder and record their lengths and average length in the table below. Place 3 cylinders in the solution labeled S1. Place the other 3 in the solution labeled S2. Allow the pieces to remain in the solution for 30 minutes. B. Remove the cylinders from the solutions. Dry them with blotting paper and measure their lengths. Record the final lengths and their averages in the table below. The table comprises 4 columns and several rows. First row, potato cylinder, original length in centimeters, length in S1 after 30 minutes, length in S2 after 30 minutes. Column 1, you have the potato cylinders 1, 2, 3, 4, 5, and 6, and then the average. C. Question 1. Account for the results in B above. That is in the table. You get 6 marks. D. Roman 1. What would happen if the potato cylinders in S2 had been boiled? And Roman 2 of D, you give a reason for your answer in D1 above. You get a mark each. Question 2. You are provided with specimens labeled J, K1, and K2, which were obtained from the same mammal. A. Name specimen J. B. Name the part of the body from which specimen J was obtained. C. Roman 1. Name the bones that articulate with specimen J. C. Roman 2. Which one of the bones named in C1 above controls or limits movement? And D. You are to draw and label specimen J. State the magnification of your drawing. Question 2E. What type of joint does specimen J form with the bone of the upper arm? F. Name the part on specimen J which increases the surface area for muscle attachment. G. Examine specimen K1 and K2. K1 and K2 are the same except K2 was placed in hydrochloric acid overnight. Describe the texture of K1 and K2. Roman 2. Name the inorganic compound that was removed by addition of hydrochloric acid to the bone. What is the significance of the presence of the substance named in G above? 4. Name the material that remained after removing the substance named in G2 above. And 5. Name the cells that are found in the material named in G2 above. Question 3. You are provided with specimens labeled L and M. Cut specimens L and M lengthwise through the middle part. Place the specimens in petri dishes. A. Roman 1. Add a drop of iodine solution to the cut surfaces of the specimen L and M. A. Roman 2. Account for your observations in A1 above. And A. Roman 3. What is the significance of the food stored in specimen L to the seed? 3B. Draw and label the internal parts of specimen L. State the magnification of your drawing. C. State 2 observable differences between specimen L and M. We now come to the marking scheme. And we'll start with equation 1A and B, where we were filling the results in the table, recording the final lengths and the averages. For potato cylinder number 1, 2, and 3, 4, 5, and 6, the lengths are 4 plus or minus 0.1. And therefore, the average of the original length is 4 plus or minus 0.1. That means the length can be 3.9 or 4.1. Length in S1 after 30 minutes for 1, 2, 3, candidates can get 4.1, 4.2, or 4.2. And when length in S2 after 30 minutes is measured, it is observed that there is a decrease, and the minimum decrease is 0.05, and the maximum decrease is 0.30. For cylinders 4, 5, 6, length in S1 after 30 minutes, length in S2 after 30 minutes, the length in S2 is 3.7, 3.7, 3.7, giving you an average of 3.7. For the length in S1 after 30 minutes, there will be increase, and the minimum increase is 0.05, and the maximum increase is 0.30, so that you can have 4 4.15 or 4.4, if I'm not wrong, and the average is 4.16. Question 1c. Account for the results in B above. S1. Lower concentration than cells up. Water enters potato cylinder by osmosis through the semi-permeable membranes, increasing the size of the cells, expanding the size of the cylinder. We have alternatives for lower concentration, like hypotonic, weak, or dilute. And increasing in size of cells, you can say increase in the length of the cylinder as an alternative. S2 accounting. Higher concentration than cells up. Hence, water leaves cells of potato cylinder by osmosis through the semi-permeable cell membranes, decreasing the size of cells. Hence, the shrink in size, or the size shrinks. Alternatives to higher concentration are hypertonic, or stronger, or concentrated. And for decreasing the size of cells, you have the alternative of hence decreasing the length of the cylinder or decreasing the size. How do you mark 1C? Accept becomes bigger or length changes from, or becomes shorter, length changes from, but reject semi-permeable alone. It should be semi-permeable cell membrane, and reject increase in cell. It is increase in the cell size. And reject the cell membrane, it should be semi-permeable cell membrane. So you reject cell membrane alone. Note, in biology, high osmotic potential means there is more water. Hence, the solution is hypotonic. Low osmotic potential means there is less water. Hence, the solution is hypertonic. High osmotic pressure means the solution has more solute, hence it is hypertonic. Low osmotic pressure means the solution has less solute, hence hypotonic. Also note, you can begin the statements by cells up, you reverse the sentences above. And don't forget to check equivalent marks in the second account. 1D Roman 1. What would happen if potato cylinders in S2 had been boiled? Answer: no change. But there are alternatives like remains the same, or no decrease in length, or no osmosis, or no loss or gain. Roman 2, give a reason for your answer in D1 above. And the answer is, osmosis is the property of living cells. Okay, or you say no osmosis. Or you say osmosis does not occur in dead cells. And also you can say boiling kills living cells. So you can accept cell died as your reason, or the cell membrane was denatured. All right. Reject non-decrease alone. It is decrease in size. Non-gain alone. It is gain in size. Destroy cell membrane. It is destroy semi-permeable cell membrane. Cell destroyed. No. Cell killed. Denature the cells. No. Denature the semi-permeable cell membrane. D2 is tied to D1. Question 2. A. Naming specimen J is a scapula. But you can accept scapula spelled with an R at the end. You can also accept scapula and shoulder blade. Accept scapula brackets shoulder blade. Accept scapula all shoulder blade. But you reject shoulder blade alone. 2B. Naming the part of the body from which specimen J was obtained. This is the shoulder. Or you can say petrogido. C Roman 1. Name the bones that articulate with specimen J. Answers: clavicle and humerus. Accept humerus if the person has put an O between R and U. And you accept clavicle if somebody has replaced CLE with CAL. But you reject humerus from the word humor, or humerus for that matter. Roman 2, the bone named in C1 above controlling or limiting movement is known as clavicle. 2D, drawing and labeling specimen J. First of all, look at what you are expected to do. For accuracy mark, you are supposed to draw the diagram proportionally. That is, there should be proportionality. There should be continuous outline and no shading. Note, position of glenoid cavity is determined by the spine. Deny drawing mark if the processes are from a rabbit. The rabbit ones are very long, but mark the labels. Also deny if two processes are labeled. A cavity as a curve, labeling to be in the curvature. Of calculation of 0.25 to 1 for the magnification, it must be correct, because calculation was not asked, but you have to be correct. So the labeling is from the top, suprascapular, spine, or ridge or projecting ridge, glenoid cavity, and coracoid process. The drawing mark is put near the rabo or below there at the glenoid cavity. Rabos, 3 marks, drawing 1 mark, total 4 marks. 2E, type of joint specimen J form with the bone of the upper arm is known as ball and socket. 2F, naming the part on specimen J which increases the surface area for muscle attachment. This is called the spine, or you can use the other ones, ridge or projecting ridge. 2G Roman 1, describing the texture of K1 and K2, and you find that K1 is hard, while K2 is soft. K1 is rigid, while K1 is soft. These are alternatives. K1 is firm, while K2 is soft. K1 is stiff, while K2 is flexible. Take care the way you give the comparisons. Mark K1 and K2 independently, each have a mark. G Roman 2, naming the inorganic compound that was removed by addition of hydrochloric acid to the bone. The inorganic compound is calcium phosphate. But you can also write the formula Ca3 bracket PO4 bracket 2, and you get the answer. 2G Roman 3, what is the significance of the presence of the substance named in G1 above? It gives bones its hardness or rigidity, so that it can carry out mechanical function. So that it can carry out mechanical function was actually not very necessary. You could just have said gives bone its hardness or rigidity. G3 is tied to G2. Deny mark if element given, but you put an omission. 2G4, naming the material that remained after removing the substance named in G2 above. It is called matrix or mesh of fibers or fibrous material or collagen fibers or collagen. Any of those ones was correct. And G5, naming the cells that are found in the material named in G2 above, they are known as osteocytes or osteoblasts. Question 3. A1, adding and drop of iodine solution to the cut surfaces of the specimen L and M. L will turn blue-black, dark blue or deep blue on the surface, on the cut surface, but reject it turns blue, or it turns black, or it turns black blue. M, you discover that on the cut surface there are little traces of blue-black, patches, spots of blue-black, or slightly blue-black. 3A Roman 2, accounting for your observations in A1 above. L has more starch, or starch is stored in L. For M, it has less or little, or you can say traces of starch. That's why the color changes the way it changes. Mark A2 if indication is blue or black alone. All right. A3, the significance of the food stored in specimen L to the seed is provides nourishment to the embryo during germination, provides energy to the embryo, provides nutrients to the embryo. During germination, it's not necessary to be said. You can just say provides nourishment to the embryo. Anyone, you get your 1 mark. But reject provides food because it is in the stem of the question. 3b, drawing and labeling the internal parts of specimen L. We will also start by the guidelines. For you to get the drawing mark, the accuracy needed is continuous outline, no shading, should be proportional, endosperm should be larger than cotyledon. Check magnification in relation to the diagram. If calculation is done, it must be done correctly. Labeling, pericarp and testa fused, scrotum, plumium, randicle, and cotyledon. How do you get the 6 marks? Label 4 marks maximum. Although the labels are 6 marks, you give 4 maximum. Drawing mark or accuracy mark is 1, and magnification is 1. Magnification lies between 2 and 9. 3c, stating 2 observable differences between specimen L and M, and L has 2 cotyledons, 2 cotyledons in specimen M and 1 in specimen L. Food stored in endosperm in specimen L and in cotyledon in specimen M. L has 2 scars and M has 1 scar. Any 2, you get 2 marks. Note, mark the first 2. For a mark to occur, there must be complete comparison. Deny color as a comparison because beans are of different varieties and maize are of different varieties. So color is not uniform due to variety of maize and beans. Asanteni sana for watching this tutorial on 2000 KCSE Biological Sciences Practical. I hope it has helped you understand the procedure and the scientific reasoning behind them. Remember, success in paper 3 comes from observation, logic, and clarity, not just memorization. Keep practicing and stay curious. Special thanks to all science educators and KCSE candidates who continue to uphold excellence in biological sciences. Your commitment to learning and teaching is the heartbeat of Kenya's academic future. The intrinsic motivation my students give me is the one which is making me produce these videos. Goodbye, guys, and may I see you in my next tutorial. My next tutorial is on biology practical from 1981 to 2000, because after 2000 we had only biology. Biology was done away, biological sciences was done away with, and we remained with biology. Those videos are already in my channel. Please watch and subscribe to my channel to get the videos from 2001 to 2017. Goodbye and good night.